A few weeks ago, I created a video on how to write a research paper. And so today I actually want to dive into one of my research papers and actually show you how I did everything that was in that video. And we're really going to deconstruct this paper so they give a really good example of how to put all these different elements together. So here's the paper I'm going to be showing you today. It is, again, a steroid and eye mobility paper. It's not the one that I typically show in my videos. Usually I'm showing my first published paper. This is actually my second published paper. And we are going to go ahead and get started in our introduction. And so you can see here that our very first sentence here, I'm going to go ahead and highlight it, is steroid analysis has become an important area of research because of the use of steroids in biological applications. And so the purpose of this sentence, and albeit I didn't, know the formula when I was writing these papers, this was still pretty early in my writing career. But the purpose of this sentence is to get your readers to understand why they should read your paper. So I'm trying to tell my reader that steroid analysis is important. So this is just basically your significance that's going on here. It's telling your reader why they should read this paper. This is a really weak significance sentence in my view. It's an important area of research because of the use of steroids in biological applications. I would have really liked to have seen some examples of this. And now, if we scroll down here a little bit, you do see that down here, we also have more significance. This is kind of telling you why this specific study is important, not the field in general. But I think this sentence here could have been bolstered a little bit more by actually sharing more examples. Why is it important? How are they used in biological applications and things like that? While I'm explaining this paper, if you're working on writing one, download my scientific research paper checklist. It's completely free and it'll be available in a link in the description below. It just helps you to kind of work step by step through what you need to do to write your scientific research article. So what you want to do is you want to take this subject here and you're going to put it as the subject of your next sentence. What you want to be doing here is simply explain what is this? What is that word here? And what that does is it makes a really, really nice flow. Steroid analysis is important, and then we're explaining what steroid analysis, or in my case, for this specific paper, I explained specifically what steroids were. This is honestly not the strongest first sentence. Steroids are uniquely characterized by 4 fused rings, and it's just kind of telling what it is. So good background information, but this isn't really important to the story. I would have rather led with this. Biologically, steroids are involved in signaling. I think that's much more important and much more relevant to the first one. So what we're entering into here is actually our background information. So all of this is just background information. Steroids are uniquely characterized. What are they doing biologically? And they can serve as biomarkers. But you can also see how what I'm starting to do is create, even within this own paragraph, a funnel. So I'm wanting my reader to go from just steroids, and then now I'm specifically talking about steroids biology. And so specifically, how are they used, and the fact that they're used as biomarkers. So then I finish up with specifically what is the impact that potentially steroid analysis can have. So this is much more of a significant statement than a background information statement. And I give those examples here: medical diagnosis, forensic analysis, and environmental analysis. So now what we're going to continue to see is that we're still diving into the background information. And what's important here is we want to be diving into the background information as it makes sense for the reader in what we're reading. So all of this here is going to be background information. So we see this is all about steroid analysis and the difficulty it presents. So that's giving them the background information. Okay, steroid analysis is important, but why is it actually worth studying? And this is why it is. This is the background information. This is the previous methods that have been used and why there are still limits here that we need to be able to address. So in the background information, we are both trying to show gap and we are trying to give the reader enough information so that they can understand what we're trying to tell them in the result. This is a really strong mix of significance and background information, actually, because it's giving the background information on the types of analysis. So I changed the highlight on this a little bit because we have a lot of good background information here, but these two sentences are really more significant. Again, we're tying in between this background information and significance to make the case for our story. So now let's move on to the next paragraph, which is all about eye mobility spectrometry. And in reality, this is basically background information. I need someone to be able to understand what ion mobility is so that they understand what I'm even trying to do here. So we're going to highlight this whole paragraph just as our background information. And then we can move on to the next one. And so what I'm doing here is I'm actually making a really nice transition between my last paragraph, which is all about background information and eye mobility, into talking about relevant literature. I'm doing it with this sentence here. Lately, IMS has been studied as a standalone separation method in combination with MS without the need for prior chromatographic separation analysis. So what I'm doing is saying, this is what eye mobility is about. And instead of just jumping into this study did this, I have a transition sentence to say, this is what it's about. And it's actually been studied in this way. So this is transitioning us into our background literature. So we have background knowledge and then we have background literature. So this one is specifically on derivatization. This one is talking about steroid dimer species. And then we have these down here that specifically talking about the previous work to this paper that I actually did, which is talking about expanding the number of metal adducts. So now we can see our three themes of our background literature going on in here. And then finally, we have a summary section. This current study examines this and what we did, and we talk about the potential limitations of this and the impact of this. So TWIM showed potential in the separation of steroid isomers and mixtures without prior chromatographic separation. So since I think introductions can be a little tricky for people to get, let's outline this up here. And let's see how is this funnel really working. So we talked about steroids, we talked about steroid biology in here, and then we're specifically going to do steroid analysis there. And then we do the different types of steroid analysis, and then we're going to go to a specific analytic technique. And so we're going to talk about IMS, which is an analysis. So you can see that these themes have been lined up in a way that we're talking about each one as they connect to each other. Then we're going to connect to previous IMS steroid studies. So that's allowing us to be able to say, okay, this is IMS. We're going to bring back in steroids and we're going to talk about what's been done before so that people understand that what we're doing is novel in this specific way. And then we go into the current study. What are we doing? And so we do a summary of the current study. And so that is essentially what is going on through this introduction. Now, there is so much more information that could have gone into this. There's so many more papers I could have talked about. In my literature review on steroid analysis and ion mobility, I talked about more papers than this. But it is perfectly fine that I didn't include all those papers to be specifically talked about in this introduction because it's not at the point. I set my start point to standalone IMS results. And there had been 4 main papers that had come out about that. So those were the 4 that I talked about to get to the point of this paper. I don't need to talk about how IMS has been used in any way for steroid analysis because that's just tangents. That's not helping my reader get to the point of their paper. So then we can move on to our method section. And my method section for this is actually very, very short, which is perfectly fine. I don't need to tell them everything. I need to tell them what they need to know to be able to do this analysis. Don't need to throw a lot of words in there. It's really simple. And whenever I'm thinking about how am I going to structure my experimental sections, I'm always thinking about going from start to finish. I don't want to be talking about instrumentation if I'm going to do a bunch of stuff before I ever touch the instrument. So you can see here, I'm starting with solution preparation because that's the very first thing that I would do. There's 2 main things going on in that section: where my materials came from and what the concentrations of my mixtures or solutions were. So then we're moving on to my experiment. And if you have animal studies, like That would be your first section generally, is how did you care for your animals? What's your IUPAC number? All of those things. So then in my instrumentation, in my actual experiment, I have a few things that I'm going to do here. So the first thing I'm going to be doing is telling them what is the instrument I used. So that is the main time I talked about the instrument. Then I'm going to tell them how I used that instrument. So all of this is telling them how I use that instrument, and it's giving them what they need to know for the important parameters that could affect this actual study. So I didn't tell them a list of every single parameter that instrument had. Instead, I focused on the ones that I knew could affect the study. So capillary voltage is really important, and it can actually depend on whether you see ions or not. The same thing with sample cone voltage and source temperature. And then these we've specifically seen drastically can change CCSs and different results we get. So that's what I'm sharing with them because that's what they need to know. And then the final thing I'm going to talk about is the types of analysis and software that I use to be able to run this analysis. And that's that component. So again, this section had 3 different components: the type of instrument, how I use that instrument, and how I visualize or analyze that data. And then finally, you're talking about data analysis. So moving through this, I prepared my solutions, I ran them. How did I actually analyze the data? And this is a little bit in this IMS and MS section in that bottom portion. And over here, it's in this one specifically in how did I actually do specific calculations. So this really just has 2 sections that I'm talking about here. One is the equations that I did for my resolution, and then the other one is how I got my CCS measurement. So then we can move into the results and discussion. And so I'm just going to give you kind of an overview here. First, we're doing ATDs for isomer mixtures. If I could go back, I would change this. I think that's a really, really not great heading. It's not information packed. It's not telling people the conclusion that they should get from this. And it's also a really, really long section. So you can see that's like over a page, probably combined here, where all my other sections are much shorter. So you can see then I talk about resolution of isomer mixtures. And so I have this figure here. And then I talk about CCSs and analysis of complex mixtures. So I'll get into each one of these sections in a minute. But in this one, I think this section really lacked direction for me. I was trying to talk about these ATDs, and I was just explaining figures and not really diving into what are the conclusions were. And so that's something that I always suggest you do whenever you're looking at your results section, is come up with your conclusions and base it off that. Whenever I first started writing papers, a lot of times my first sentence, I didn't know what to say. And so I would just say like what was presented in the figure that I was about to talk about, which is a way, I mean, this got published, like it's definitely a way you can do it. But one of the things you might want to think about is referencing a method instead. So I could have said, IMS analysis of mixtures showed that certain steroid isomers maintain separation as seen in a single sample, while others lost that separation. And I think that would have been a much better first sentence. And then I could have separated up these headings into isomer pairs that maintain separation and isomer pairs that lost that separation. And so you can see here that I think these first 2 I could have maintained, that they maintained their separation and specifically talked about them more. And then down here, this could have been its own actual section as well. So we could say like or testosterone isomer analysis. I think this section was really long and really lacked direction. You can see that just in, I'm basically just explaining the figures here. So I'm going from one sample to another, like this specifically. Like in my case, we do results and discussion together. I would have rather seen this as a full paragraph. Additionally, it was noted that corticosterone had a lower ionization efficiency than this one in all dimer addicts, resulting in a lower signal at the same concentration. My scientist brain now says why. Like, I want to know why. You're not telling me why is that the case? What are the impacts of that? And is that what's been seen previously? It's just saying it here. So I would have rather separated out this incredibly long paragraph and dived into each one of these, answering the why and the impact for each of these. And so that's something that I really didn't do well, which you can see, like, this got published. You cannot have the most perfect paper in the world and still have a published paper. We can see that I am bringing in some comparison to previous analysis here. So you can see that I'm saying that they were baseline separated in Reister et al., which is the first paper I wrote. However, in this one, they formed a heterodimer. So that's kind of why is it different than before? That's one of the things I talked about in that video is you need to explain why is it different than before so that people understand yours in the context of previous literature. So I think in some places I am doing a really good job in the discussion here, and some places I'm just not, unfortunately. And so I think this could have used a tiny bit more time. If I would go back now, I would definitely change certain things within this. So here's again me comparing to previous literature showing that this actually did better than a 7-minute method when this was essentially a 1-minute method. So you can just see that's what I'm trying to move to, is trying to show, hey, are we doing better? Are we doing worse? Why are we different? All of those things. But I think what I really lacked in this discussion was a good understanding of why are all of these things happening? Why are some things close together? Why are some things far apart? One place where I did talk about the why is in here. So I say that they were the only steroids that could be separated as a monomeric. And then I say this is likely due to their more complex structures among the steroids separated. Then we move into this one, which is resolution of isomers. And so again, you can see this first sentence here. I'm just telling you where to find the information. I said were calculated for the separation according to equation 1. So that's at least a reference back to the method section, which I think is important to have in your results section. So that's referencing you to the supplementary information. And then over here, I like that I did one sentence here. So it starts here, most isomeric pairs had a resolution of 1.0 for at least one addict, the only exception being estradiol isomers. So that's a really good impact thesis sentence. And so I think this is actually a really good example of doing a results and discussion. So we have this sentence, which is doing directions. So where to find information. Then we have a really good thesis sentence that I'm highlighting in gray. So this is all discussion right here. So the only results that I gave is this, because mainly I was always giving resolution values back up in the ATDs. I'm sharing all of this up here. So this is mainly discussion of the resolution. It's why did these get mistaken? There's the heterodimer in there. There's other complications and different mass to charge values and all of those things. And then I give a really good conclusion sentence for what this is related to. Now, I wish I had said this in much more plain English, but basically because of the isobaric nature, some of these can actually start to overline with each other, which might make things a little bit more difficult. So you can see that the next section is CCSs, and you can see that overall, I'm trying to show the error that's involved in here. So you can see I am telling them the relative error for the mixtures was below 2%, which is what is considered valid in this field. And then saying previous analyzed could be used in identifying steroids. So that's the impact. And then finally, we did a complex mixture. So we added them all together and separated them out. So my first sentence in this is all about objective. So this sentence is telling them why we did this analysis, which is what I suggest other people to do, is to have an objective or a methods section telling you how you got these results or why you got these results. And then I share the results for this. So we can see all of this is results. And then what we did is I actually had a section dedicated to the discussion, right? Like, why is this happening and what are the impacts of this happening? But I'm also talking about the fact that there are mixtures of dimers going on here and that you may not get good quantitative analysis because of this. With the results and discussion done, we finally have our conclusion section. And so I actually started out this conclusion section, which is kind of interesting to me. I don't remember ever doing this. Usually I start out a conclusion section with significance again. In this case, I didn't. I started it out with a comparison. And so I say previous works illustrated the enhanced separation of this in single component standards. I wish I would have had a significant sentence above this. I think this is an awkward place for people who aren't reading my paper to, whenever they just go to their conclusion sections, starting with this sentence is a little awkward, and I don't like it now that I'm like reanalyzing it. So then I have a really good sentence here that is the impact, the significance of the study, which I would recommend doing. You can have significance of your field and significance of your study. And then I'm talking about the main findings here. So this is all of the main findings and significance of this. And then we're going to jump into limitations. So this is some limitations here. It can hinder the ability to quantitate and the resolution. And then we're jumping into impact and future directions. So impact is this sentence here, and then future work. And guys, please include future work whenever you're writing research papers. It's like, you should know what's coming next. Tell people what's coming next. So overall, I don't like this sentence, but there's a significant sentence here. This is a mix of results and significance. And then we have results here, one sentence on the limitation of this, an impact sentence, and then a future work sentence. So overall, I hope that this helps you understand what is really going on in a research paper, mistakes that I made that you can maybe prevent making, but in reality, it can still get published even if it's not a perfect paper. If you want to learn kind of much more simply how this whole process works, check out this video here all about how to write a research article, and you can also check out my scientific communication playlist down here. If this video was helpful, like it and subscribe to the channel if you want more videos on how to become a more efficient researcher. I hope you enjoyed this video, and I look forward to seeing you in the next one.